I. B. Jalakshmi, Botany Department, Jamshedpur Women's College, teaching you the topic glycolysis from the chapter respiration. The topic is for the student of Sem 6 Botany Honours. The process of glycolysis is common for aerobic respiration and anaerobic respiration. Therefore, it is regarded as fundamental step in respiratory breakdown of glucose. The sequence of reaction which convert glucose into pyruvic acid with the production of ATP is termed as glycolysis. The steps of glycolysis were studied by three German scientists, Embeden, Meyerhoff and Parnas. On their names, the glycolysis is also called EMP pathway. The steps of glycolysis takes place in cytoplasm, and through glycolysis, glucose is oxidized into pyruvic acid in the presence of many enzyme and coenzyme present in cytoplasm. The oxygen is not required at any step during glycolysis. Glycolysis can be divided into three stages. The first stage is phosphorylation of hexose sugar. Number 2 is splitting or cleavage of fructose 1,6-diphosphate, and number 3 is formation of pyruvic acid. Altogether 10 steps are there. The synthesis of fructose 1,6-diphosphate takes place by phosphorylation of hexose sugar, which combine with phosphate to form different types of hexose phosphate. The attachment of phosphate with hexose sugar is called phosphorylation. During phosphorylation, ATP plays an important role by acting as phosphate donor, and it is converted into ADP. The first step of glycolysis. The phosphorylation of glucose molecule takes place, in which glucose molecule react with ATP to form glucose 6-phosphate and ADP. That means in the first step, glucose is converted into glucose 6-phosphate. Now, the step 2, glucose 6-phosphate undergoes isomerization in presence of enzyme phosphohexoisomerase to form fructose 6-phosphate. That means step number 2, glucose 6-phosphate is converted into fructose 6-phosphate. Number 3, in this step, further phosphorylation of fructose 6-phosphate takes place with the help of ATP, resulting into formation of fructose 1,6-diphosphate in presence of enzyme phosphofructokinase. Here, in these three steps, phosphorylation of hexose sugar is completed. Now, the second stage, splitting or cleavage of fructose 1,6-diphosphate. Fructose 1,6-diphosphate formed at step 3 in stage 1. Now split into two molecules of 3 carbon compounds, 1,3-phosphoglyceraldehyde and dihydroxyacetone phosphate with the help of enzyme aldolase. Both these compounds are called triose phosphates and are interconverted into each other in the presence of enzyme phosphotriose isomerase. Now the further reaction, two molecules of 3 carbon compound will be involved. Next step is formation of pyruvic acid. The step 5. In this step, 3-phosphoglyceraldehyde react with phosphoric acid H3PO4, resulting in the formation of 1,3-diphosphoglyceraldehyde. Step number 6. 1,3-diphosphoglyceraldehyde is oxidized into 1,3-diphosphoglyceric acid in the presence of enzyme diphosphoglyceraldehyde dehydrogenase and a coenzyme NAD. During this reaction, two molecules of hydrogen are released from diphosphoglyceraldehyde, which reduces NAD to NADH2. Step number 7. The diphosphoglyceric acid loses one of its phosphate, which combine with ADP to form 3-phosphoglyceric acid and ATP in the presence of enzyme phosphoglycerokinase. This step is important because ATP is formed here. Number 8. In this step, an isomeric change takes place in 3-phosphoglyceric acid, resulting into transfer of phosphate from carbon 3 to carbon 2 and formation of 2-phosphoglyceric acid in the presence of enzyme phosphoglyceromutase. Number 9. 2-phosphoglyceric acid loses 2 molecules of water in presence of enzyme enolase and converted to phosphoenol pyruvic acid. Now the step number 10. This is the last step of glycolysis. Here, one phosphate group is removed from phosphoenol pyruvic acid with the production of pyruvic acid under the influence of enzyme phosphopyruvic kinase. The removed phosphate group is accepted by ADP to form ATP. So the last step is very important. Now the summary of glycolysis. In glycolysis, one molecule of glucose, from one molecule of glucose, 2 molecules of pyruvic acid are formed. Number 2. In this, 4 molecules of ATP are formed at step number 7 and step number 10. Because 2 molecules of ATP are consumed in phosphorylation reaction, that is at step number 1 and 3. Therefore, during glycolysis, there is a net gain of 2 ATP molecules. 4 ATP minus 2 ATP is equal to 2 ATP. The last point is 2 molecules of NAD are reduced to 2 molecules of NADH2 at step 6, which later on oxidized aerobically to yield 6 molecules of ATP. Because one NAD molecule after oxidation produces 3 molecules of ATP. Thus, the total gain of ATP molecule during glycolysis in presence of oxygen will be increased to 8 instead of 2. Now this chapter is finished.